Hey, this is Miss Reynolds, and I just wanted to record for you an example of the microscope. I know that you have this on your virtual lab. However, I wanted you to be able to see this in person. I've got two different microscopes here. They're both protected by these dust covers. It's always a good idea to keep your microscope safe from dust, and they'll last longer. This one here, this larger one, is a stereo microscope, and I have a special eyepiece on here that enables it to go up to about 50 times magnification. And so there's 4 times, 5 times, and then with this added piece, 50 times objective multiplication. And so I can look under here and see things 3D. I can look at bugs and insects, worms, leaves, all that sort of stuff, things that are larger and macroscopic that you can see with the naked eye. You can even put coins under here. I really like this microscope. I just got this one. And this other one here is the traditional bright field light microscope. And this is the one that's used in labs and in the classroom. So I'll give you a tour of my small home lab. And that's because I'm a microbiologist, so I love doing this. I just love looking at cells and looking at things under the microscope. So I have some things at home so I can look at them. I have right here a low-speed centrifuge. These are my lab glasses. I've got a lab coat and gloves as well. This is a slide warmer. I have a hot plate coming as well. I have a scale. I have a vortexer and laboratory glasswork, a small little sharps biohazard container, test tubes, large ones. These are your typical plastic types that you can write on. I have stain kits, slides and cover slips. Believe it or not, you can look at the underside of a leaf just by adding some quick clear nail polish to the bottom of it and then peeling it off, putting it on a slide, and you can see the little pores where they take in the molecules from the sun, CO2, and then where they put out oxygen and water. So I'll show you those. These are just different stains. I have a gram stain kit in here. That's a differential type of stain. I have the simple stains. I've got some Lugol's iodine, so I can show you some experiments with that. This is an acid fast stain kit. I have a right stain for blood, and this is another simple stain, a red one, and I've got the blue one, the methyl blue. This is Resolve oil for oil immersion, and this is Safranin O solution. You can look at fats with that, or you can counter stain organisms with that as well. And then somewhere around here I got the methylene blue, and believe it or not, that is the same dye that's in the blue toilet stuff, those tablets you drop in, or the cleaners, because it has antimicrobial properties. So that's why they're blue. This is a microscope, and this is the eyepieces. The eyepieces are called the oculars, and they are adjustable depending on the width of your eyes. Each person is unique, so this will be different for every person. The eyepieces come out, and each one tells you how much multiplication they have. This one is 10 times. They both are, so 10 times. And then you have the objectives, which are on a nose piece. It turns and clicks and locks into place. So each one is color coded. I'll start with the lowest magnification. This one is represented by the yellow band right here. It tells you on the outside, it's carved into the outside, which objective lens this is. On mine, this one is 10 times. So that is also called the low power objective. So this is the one you start out with when you want to focus the microscope. You then go over to this one. Not every microscope has this one here. This one is unique. It was purchased special for this. This is a 60 times objective lens. This one though, you cannot use oil immersion on that one. And you cannot use it on this one either. This one is my high dry, which is my 20 times lens. Usually instead of the 60, this is a 40, but this one is a little bit higher power. This one's 20. Again, you're high and dry. You want to move to that one next after you get your cells in focus under the 10 times objective. And then the white band, this one is the 100 times oil immersion objective. You only use this one with immersion oil, and clean it well before and after use, because we don't want to scratch the lenses. And never, never, ever swipe any of the other objectives through the oil, because it will damage the lenses. So this one is the one that will enable you to see the most and see the most detail under the bright field light microscope. The back here is the arm, the handle. This is how you carry the microscope. Most microscopes are a little bit heavy, a little bit awkward, and you want to support it by the arm and put the other hand under the base. The base contains the light bulb. You have to turn this entire thing over when you want to change the light bulb, the light source. And it comes up through here, through this special type of diaphragm condenser. This is how you turn the light source on and off, and it's adjustable by this knob here. You can turn it down or up. You can see that it's on, and you're able to turn this a little bit. I always clean this to make sure that it is dust free, because the light comes up underneath here first and hits the specimen first up through the bottom. So you can see that's where the light's going to hit the specimen on the slide. This piece here is the stage. The stage clips are what hold the slide into place. Believe it or not, you can remove this entire thing. You can move the stage clips, lower the stage, and you can put an entire petri dish on here and look at things growing under the microscope on the stage without having the clip on here. You can make the light go up and down right from right here as far as how it's hitting underneath the specimen with this aperture here, the condenser, and this is the diaphragm aperture, which you can kind of turn this into phase microscope for things that are transparent like yeasts, things that are hyaline that you can see through by turning this down. You can also lower this completely when you want even less light to come through. So that lowers that up and down. You also have the coarse knob and the fine tuning knob. The coarse knob is going to make the stage go up and down, and this is how you first bring things into focus. So you want to work with that a little bit at a time. Once you get something in focus and you want to fine tune it, you turn this knob right here, and there's one on both sides of the microscope itself. So these come out, these come off, and you can actually take them off to clean them every now and then with a sterile swab and some rubbing alcohol to keep these nice and clean. You get the total magnification by multiplying your ocular, which is 10 times. You only use that once because this is considered one field of view. So even though each one of these is 10, you only count them as 110. Okay, so 10 times whichever one of these objectives gives you your total magnification. So my 10 times my 100 objective is going to give me 1,000. My 10 times the 20 is going to give me 200 total. My 10 times is going to give me a total magnification of 100. And then finally, my 10 times 60 is going to give me a total of 600 magnification. So that is total magnification. And when I'm finished, I'm going to turn the light off. If I had put a slide on here with oil, I'm going to clean it off and make sure that it is nice and clean, and I'm going to cover it back up with the dust cover. I hope if you have any questions, you'll reach out to me, but that is just an overview of the bright field light microscope. And this is the stereo microscope. The stage does not go up and down, but this entire eyepiece does. My one objective is built into this. You can buy different magnifications to put on here, but this one enables it to be, it's a 50, so I have a total of 500 multiplication with this. This moves up and down this way, so the whole head piece moves. We have not hooked this up yet, but we have the ability to hook up a camera on here. That's in the works. There are slide clips here. This is a dark background, but I also have a reversible type, so you can look at it on the light background or the dark background, depending on what you're looking at. The light source is this right here, so it comes from the top and it's 3D as opposed to coming up through the bottom of the sample. And again, this is just great for things that you can already see with the naked eye. That would not work under the bright field microscope, but you can get great 3D detail of insects, of leaves, coins, salt particles, sand particles, all that sort of thing with this type of microscope. I hope if you have any questions, you'll reach out to me, and I look forward to working with you. Okay, have a great day. Bye.